Assalamualaikum and good day everyone. So today you will be learning about chemical structure and biological function of lipids and membrane. So lipids, they are characterized on their solubility behavior. So lipids are hydrophobic in nature because they are soluble in non-polar solvents. For example, alcohol, diethyl ether, methanol, and hexanes. When we are talking about hydrophobic, meaning it is phobia towards water, so lipids, they are not fully solubilized in water. So we can identify or characterize lipids from their functional groups. So lipids have carbon-carbon single and double bonds, aldehydes and ketones, carboxylate esters, phosphate esters, amines, and amides. So the main role of lipids are as a long-term energy storage. So the main structure of lipids are fatty acids, which is the components of non-polar lipids, or we call it as fats. So these fatty acids are the important energy molecules in heart, brain, and adipose tissues. So the lipids is also present in the form of biological membrane, where their structure are considered as polar lipids containing nitrogen and phosphorus, where the composition of biological membranes are containing lipids and proteins, which make them as a molecular boundaries around all cells and cell organelles. Other types of lipids is steroid, which is it represented in the form of cholesterol, which is also found in membranes and also serve as precursor for many hormones. So in this topic, you will learn several categories of lipids based on their function. So the first, we will look on the energy storage, the lipids in the form of triacylglycerol, or we call as fats. And then we look on lipids in the form of membrane structure. So there are several types of lipids, which is phospholipids, sphingoglycolipids, and cholesterol, which is make up the membrane structure. And then we will look on the category of emulsification in the form of bile acids. And then lipid as messenger molecules in the form of steroid hormones and eicosanoids. Lastly, the protective coating as a biological wax. So this is an example of the lipid structure. So you can see lipids are different in structure based on their biological function. So the first on the left-hand side on top is a fat in the form of triacylglycerol, or we call it triacylglyceride. In the middle is a biological wax. And the right-hand side, most on the upper is a steroid, right, where we can see a cholesterol structure. And at the bottom is a glycerophospholipid. Bottom in the middle is sphingophospholipid and sphingoglycolipid, which is made up of the cell membrane. So a lot of these structure represent lipids in different form, but basically all these lipids are insoluble in water and they are soluble highly in non-polar solvent. So first we look on the first structure or the first type of lipids in the form of triacylglycerol, or we call it as non-polar lipids. So we call it non-polar lipid because it has very hydrophobic region, which is served as storage molecules for the metabolic fuel. So the structure will be in the form of fatty acid, right, which is found as a constituent of the triacylglycerol, where the fatty acid bind to the 1-glycerol at the hydroxyl group of the glycerol through the process called esterification. So we will see the different type of fatty acid, whether it is in the form of fats or we call it as a solid triacylglycerol, usually isolated from animals, which it contains predominantly saturated fatty acid. And another type is in the form of oil, where it is in the form of liquid triacylglycerol, usually isolated from plant seed, where it predominantly as unsaturated fatty acid. And lastly, the structure of triacylglycerol is in the form of wax as a protective coating for plant leaves, or beeswax, and ester composed of palmitic acid, which contain 16 carbon without any double bond, and alcohol tricontanol, the saturated chain, which contain 30 carbon. So this is to show an example of wax which have large molecular weight, where the structure having a esters. So R is number of carbon, right? So several example of wax we can see in the birds or insects, which as a water repellent on the feathers or exoskeletons of the insects, or on the leaves or fruits, which can help to minimize water evaporation, or in human as a wax that we can see in our ears, right? So for example, below beeswax, it has the R or the carbon 15 and the R prime carbon 30, right? So another example is carnauba wax, or we call it Brazilian palm, which contain the R structure of 31 carbon and R prime carbon 34. And another example is in the whale, the head of the whale, which contain 4 tons of this wax, which is help the wax to dive to feed the for feeding, which it can help to increase its density, right? So that the whale can stay submerged without effort. So until they are ready to return to the surface, so the oil or the wax will be remelted to help them to buoyancy. So this is a picture, an example of beeswax. And next it is a structure of a wax containing palmitic acid, right? Dying with the tricontanol structure. So we move on to look on the structure of fatty acid. So basically, fatty acid contains carboxyl group, COOH, that connected to unbranched aliphatic chain. So fatty acid usually as a monocarboxylic acid. So their differences is based on the number of carbon. So the longest basically is 36, right? So here is 12 to 26. This is an example. So medium is around 8 to 10, and the shortest is 4 to 6. And this fatty acid is considered amphiphilic because it has one end, which is polar or ionic, which is the carboxyl group, and the opposite end, which has a hydrocarbon chain, which is the non-polar group, right? So the bound in fats, triacylglycerol, and other lipids. So it has a hydrocarbon chain, which number of carbon atoms range 4 to 36, which is I said earlier, it is unbranched. And if it is saturated, it contains carbon-carbon single bond. And if it's unsaturated, it's either mono unsaturated containing one double bond, or poly unsaturated fatty acid, which has 2 or more double bonds, usually which is less than 6. And in nature, the fatty acid of this unsaturated fatty acid are in the cis configuration. So now we look on the structure of fatty acid. So fatty acid is actually an organic acid, which is have a chain of carbon with hydrogens that attach to it, that has an acid group at one end, right? C double bond OH, and methyl group at the other end, CH3. So this is an example of triacylglyceride. So where this is the structure of the glycerol, where 3 fatty acid, for example, combine on the first OH of the glycerol, the second fatty acid, and the third fatty acid attached to the OH of the glycerol. So the triacylglycerol formation is actually through condensation, where glycerol will be first bind with the first fatty acid to form monoacylglycerol to release water, and then monoacylglycerol bind with another or the second fatty acid to form 1,2-diacylglycerol to release the second water molecule, and then the 1,2-diacylglycerol react with the third fatty acid to form triacylglycerol to release the third water molecule. The fatty acid, they are vary in length, which can affect the absorption, and also the saturation, which the chemical structure can affect the cooking and also the storage properties as well as human health. So the fatty acid, if it's saturated, so it basically have carbon chains with hydrogen atoms without double bond or no double bond. So saturated fat, or we call it triacylglycerol or triacylglyceride, containing 3 saturated fatty acids, such as in animal fats, for example, butter and lard, and also tropical oils such as palm and coconut. And usually saturated fatty acid, they appear in the form of solid at room temperature. This is an example of saturated fatty acid containing 18 carbon, which call it as stearic acid. So you can see there is a methyl N and also the acid N of the stearic acid. So you can draw either this kind of structure or below. So this is the simplified structure. So another type of fatty acid is unsaturated fatty acid. So the carbon chain lacks some hydrogens, which they have at least 1 double bond. So monounsaturated fat triglyceride containing fatty acid with 1 double bond, for example, canola and olive oil, or polyunsaturated fat triglyceride that contain high percentage of fatty acid with more than 2 double bonds, for example, like corn, safflower, soybean, sunflower oils, and there's some fish oil that contain polyunsaturated fat. And usually unsaturated fatty acid, they are in the form of liquid at room temperature. So these are example of 2 unsaturated fatty acid which contain the same number of carbon, 18 carbon. So the upper one is called oleic acid, which have only 1 double bond. We call it monounsaturated. And below is linoleic acid, which contain 18 carbon, which have 2 double bond, right? Or we call it as polyunsaturated fatty acid. So this is just to show the structure of fatty acid. the left hand side is saturated, right? And on the right hand side is a structure of fatty acid, which has one double bond. So you can see if it's in the form of this ball structure, so you can see that it looks like a bend or bending due to the position, or we call it cis configuration. And these are example of other fatty acid. The first one on top is saturated, or we call it stearic acid. So monounsaturated, or we call it oleic acid, which with one double bond, and polyunsaturated linoleic acid, which contain two double bond, right? So all these are basically containing 18 carbons. So there are also various formulas that can be used to draw the long chains. For example, like myristic acid, 14 carbon in long. So you can draw either like the common structure, CH3, CH2 until the end, or you can draw like a zigzag, or you can make a short or abbreviation or the name of the structure of the fatty acid. So the structure notation for fatty acid. So for example, the first one, saturated fatty acid without any what we call this double bond. We use this triangle, right? The notation. So without the notation on the acid end and also the methyl end. For example, the monounsaturated, usually they have what we call this, the double bond at position carbon number 9, and usually it has methyl end with omega 3 or omega 9, while the polyunsaturated fatty acid usually position at carbon number 9, 12, and 15, with also methyl end of omega 3 and omega 6. So fatty acid, for example here, palmitoleic acid. So the double bond in most unsaturated fatty acid are in the cis configuration. So if there is more than one double bond, so they occur at 3 carbon interval. Right, for example here at carbon number 9, right? 10, 11, 12. You see, so you have to see that the occurrence of the double bond must have 3 carbon interval. See 1, 2, 3. So meaning at the first double bond you will see at carbon number 9, and then the second will be positioned at carbon number 12, right? So this is divinyl methane pattern showing that the cis configuration. So double bond pattern in the polyunsaturated fatty acid, right? So cis pattern, so it creates the band molecule. All right. So the space filling models of 418 carbon, you see, of fatty acid which differ in the number of double bonds will be presented below. So you have to note how the presence of double bond change the shape, right? So this is the first one on the left on top. It is without any double bond, right? On the right hand side, it has a one double bond. On the left below is 2, and on the right hand side below there are 3 double bond of this 18 carbon fatty acid. So the fatty acid component in oil, they are differ. So to know that the oil are good on bad oil, so usually we will consider on percentage of saturated and unsaturated fatty acid. So if you can see from this table, so the most oil that have unsaturated fatty acid are the canola oil, right? So the second will be sunflower oil, right? Which has the percentage of unsaturated fatty acid. And then you can see that the least best of oil contains saturated fatty acid, right? So which you can see the most that is not good oil you can consider is coconut oil, right? Because it has the most percentage containing carbon 4 to carbon 12, 60% of saturated fatty acid, right? So this is another type of uh, figure or table to show that the coconut oil, for example, have uh, more saturated, and then the second one followed by mono unsaturated and polyunsaturated. So the more saturated fats that present in the samples, meaning it's bad, it's not a good oil or lipids for health, right? So we can see that the most least saturated fat can be present in cottonseed oil, right? So this is to show some example of oil. For example, here is canola, or it actually comes from the name Canadian oil acid. It is from Canadian rapeseed, where it has the ideal ratio of omega-6 to omega-3 fatty acid 2 to 1. So it is considered as good oil because of they have low saturated fat. So next is olive oil, which is deemed to be the most healthy, which is it has the highest percentage of mono unsaturated fatty acid, and the most or the best olive oil we consider as the first press oil, or we call it as extra virgin olive oil. And this is to show you the structure, names, and common name of fatty acid. So we look on the first one, number of carbon 12. We call it the common name for carbon 12 is lauric acid. The systematic name is dodecanoic acid. The abbreviation is 12 colon 0. And the structure is CH3, CH2 in bracket 10, COOH. So this is to show you how the names of the fatty acids are being the formula. So usually it's either we use the common name or systematic name. The common name is the one that usually being used worldwide to represent the number of carbon, right? For example, like carbon 14 is myristic acid, which is considered saturated fatty acid. Palmitic acid, 16 carbon, also saturated. Palmitoleic acid, carbon 16, right? And then stearic acid, 18 carbon. Oleic acid, 18 carbon with one what this double bond at position carbon number 9, right? And then you can see linoleic acid is also 18 carbon but has two colon 2 mean it has two double bond position at carbon number 9 and carbon number 12. And linoleic acid, you can see 18 carbon with 3 double bond position at carbon number 9, 12, and 15. So as you can remember just now, the position of the double bond must have interval of 3 carbon. And next we can see on the other type of fatty acid. We have 20 carbon, we call it arachidonic acid. You see there are 4 double bond, right? And then I'm sure you have heard EPA and DHA. It is essential fatty acid, which is 5 and 6 double bond. All right. So in order to understand about the naturally occurring fatty acids, so you need to know the general rules of the structures. So first, most of the fatty acids have an even number. So there are no fatty acid with odd number. Usually there are 4, 6, 8, 10 carbon, right? So number 2, the second rule, the hydrocarbon chain is almost always unbranched. So it's not similar like the carbohydrate. It has branches. And then number 3, most carbon-carbon bonds are single. However, fatty acid may contain 1, 2, or more carbon-carbon double bonds. And the double bonds are mostly cis configuration, right? Then the fifth rules, the monounsaturated fatty acid, the double bond is usually between carbon number 9 and 10. And lastly, rule number 6, if more than one carbon-carbon double bond is present, they are not conjugated, but are separated by methylene unit. Right? So this is a list of more representative fatty acid that usually present in our diet or in our cooking oil. So you can see the structure on the upper part, right? Below is their name, right? And their melting point differ according to the number of carbon, right? So less or shorter the number of carbon, the melting point is, so it's increased, right? When it's due to the saturation. And then the oleic acid, linoleic acid, and linolenic acid, when the polyunsaturated or the number of unsaturated carbon are more, so the melting point is lesser. Right? So the common name, so you derive from a common source of compound, which is where it was first isolated. For example, like palmitic acid is actually the first isolated from palm oil. Right? So then the oleic acid is the first fatty acid that is isolated from olive oil or oleum. Next is stearic acid, which is from the Greek word of solid. Acid is the solid at room temperature. And lastly, the spiders arachnids, which is containing arachidonic acid. Right? So just to share with you the arachidonic acid structure again. So it has a 20 carbon like this, and it has a considered a cis in structure, right? So make it like a clip, right? Bending structure of 20 carbon fatty acid. And basically this arachidonic acid is present in our blood vessel, which is involved in vasoconstriction and vasodilation of our blood flow. So during a fever, or if we infected with diseases, so usually we get fever that cause shivering, right? Or we feel pain at our muscles. So basically it's due to the production or release of the icosanoid, which is prostaglandin, the PGG2, PGH2, PGE2, and PGD2, which is catabolized by the cyclooxygenase enzyme towards the arachidonic acid. So the other pathway that involve of the arachidonic acid is thromboxane synthase, the another enzyme which catabolize this prostaglandin H2 to thromboxanes. So basically, this is where the painkiller are being developed, right? Which is targeted on the cyclooxygenase enzyme or COX, where it the drug is developed to inhibit the synthesis of prostaglandin by inhibiting the cyclooxygenase. So just want to share with you, this is a drug that already being developed as a painkiller. We call it Aleve, right? So I'm sure you have heard aspirin, or we call it acetylsalicylic acid. So it is an example of painkiller, right? Or indomethacin, ibuprofen, acetaminophen, and the naproxen. For just now, the brand name is Aleve, but the generic name is naproxen. So the IUPAC system of carboxyl carbon is number 1, right? So all bonds reference the carboxyl carbon, right? So example, double bond between 9 and 10 is the carbon 9, a Greek name. So we call it cis 9 hexadecanoic acid as a 16 carbon. Okay, so these names are awkwardly long, so we seldom use, right? So in IUPAC system, so for example here, palmitic acid will contain 16 carbon. We call it hexadecanoic acid, right? The nomenclature of fatty acid. So stearic acid is an octadecanoic acid, 18 carbon, right? So oleic acid, 9 octadecanoic acid. So it has one double bond located at carbon number 9, right? So linoleic acid and linoleic acid and so on, right? So this is, we call it trivial name and the IUPAC name. Okay, just to show you how the structure looks like, right? Differences when they have a double bond and without. So there are simple reaction of fatty acid. So basically, the fatty acid will be metabolized in our system in the presence of oxygen, right? To produce several pieces of carbon-carbon structure to release carbon dioxide and water to release energy. So basically, we can metabolize fatty acid in our body system, but some fatty acid we can't synthesize in our system. So that is called as essential fatty acid. So which we need it through diet. So the essential fatty acid are linoleic, which is 18 carbon with 2 double bond at position 9 and 12, as well as linoleic acid, which is also 18 carbon with 3 double bond, 9, 12, and 15. Right, so essential fatty acid, I'm sure you have heard ALA, alpha linolenic acid, which is an omega-3, and linoleic acid, the omega-6. Right, so we need those omega-3 and omega-6 from diet because we can't manufacture it in our body system, right? So when sufficient quantities are supplied in the diet, the body can make enough, right? These docosahexaenoic acid, DHA, and eicosapentaenoic acid, EPA, or we call it eicosanoids. The one that I shared with you just now, the prostaglandins, right? It is the eicosanoid, right? Yeah, so basically the ALA and linoleic acid has a relationship with the function on our brain development, where if we lack of omega-3 and omega-6 in our diet, it can cause depression, ADHD, Alzheimer's, and also cause an inflammation. Right, so fatty acid. So location of double bond, omega number, so it refers to the position of double bond nearest the methyl end. Okay, so how this omega-3 fatty acid and omega-6 are named. All right, so this is how, for example, linolenic acid, the omega-3 fatty acid. So we consider the number of carbon from the methyl end, carbon number 3, which has the first double bond, right? while the linoleic acid we consider as omega-6 because of the first double bond that nearest with the methyl end is at carbon number 6. All right. So mainly 20 and 22 carbons polyunsaturated. And the first in is the 3 from the omega end. So icosanoid or icosapentanoic acid, it has 20 carbon. You see, they have several double bond, right? And docosahexaenoic acid, DHA, have 22 carbon with more double bonds here, right? So, and then we have another term which has a COOH of the acid, we call it as the alpha N. And then the other side, which is nearest with the methyl, we call it omega N, right? So just to share with you the source of EPA and DHA, or the essential fatty acid that we needed, usually come from fish oil, right? So omega-3 fatty acid, which can help us to prevent from heart disease, right? Which is mostly come from fish oil, for example, like cod and herring, which they have a lot of polyunsaturated fatty acid, right? So if some of us don't like fish, so we can also get omega-3 from eggs, right? So these are the what we call this example, the list of what we call this food that containing omega-3 in seafood. So the highest in salmon, right? Around 2.6 of omega-3 per 100 grams, right? Followed by herring, anchovy, and then the least is oyster with 1.4 per 100 gram of omega-3. All right, so omega-3, how it can be present in the eggs. So basically, the chickens who lay eggs are being fed with flaxseed, which contain high of levels of this omega-3 fatty acid. So after the 10 days, this will modify the fat content of the egg yolk so that it will contain omega-3 fatty acid and less saturated omega-6. Right, so currently there is a research that is ongoing where the omega-3 meats can be developed, right? Where from chicken, cow, and lamb meats to obtain high omega-3 fat contained through and research. So other than meat, there is also a tree enriched product in orange juice and also other fruit juices, right? For example, avocado. So avocado have been identified as one of plant that contain high omega-3, right? Other than the vitamin E, antioxidants, and beta testosterone, right? And there are other vegetables or fruits that have high omega-3, the essential fatty acid, other than the flaxseed. They are high in navy beans, in walnuts, in the pressed olive oil, the kidney beans, the pecans, the salmon, the leafy greens, and also the winter squash, right? So these omega-3 basically are most important, which they can bring balance to our hormones, which can reduce inflammation, that can regulate our blood sugar, and also prevent blood clotting, where it can keep our cholesterol and triglyceride in balance, as well as to relax our blood vessel and make our cell healthy and resilient. So that's why we need omega-3 in our diet. So next we look on the fatty acid reactivity. So basically there are several reactions that involve in lipids. The first is oxidation. Second is ester hydrolysis through saponification, where the hydrolysis of ester by the sodium hydroxide to produce glycerol and soaps. Lipase enzyme, which is also undergo the ester hydrolysis, where it present in intestine and also in fat cells or we call it adipocyte to release fatty acid for energy metabolism. So fatty acids also undergo hydrogenated through addition of hydrogen to unsaturated fat, which make them more solid or firm. So it affects stability and protects against oxidation, more shelf stable. So it also widely used by food industry in margarine, shortening, peanut butter, baked goods, and snack food. And lastly, fatty acid also involve the partial hydrogenation, which used to reduce the number of double bonds in the fatty acid to change the oil into a firm but spreadable solid like soft margarine, right? So the first reaction is oxidation of the organic compounds. So for example, the fatty acid saturated, when it's oxidized, so it's usually difficult to be oxidized of the saturated fatty acid. However, the unsaturated fatty acid can be easily oxidized, right? And this oxidation can cause resinity, right, of the unsaturated fatty acid. So this oxidation, which actually is can be inhibited or cannot be occurred in saturated fatty acid, where that's why the saturated fatty acid are more stable and they have longer shelf life as compared to unsaturated fatty acid, where it is faster deteriorate, right? And to prevent the deterioration, usually we need to add antioxidants to protect the compounds in the body. Okay, so the next reaction is saponification or hydrolysis. So basically, hydrolysis means the reaction must occur with the presence of water. So saponification means the addition of salt of sodium hydroxide, where it will react and release the single structure of glycerol and release the three fatty acid with the sodium bind to form a sodium salt or soap, right? Another reaction of hydrolysis by the lipase enzymes. This is occurred in our intestine, where the triglyceride that occurred either in intestines or in our adipose tissue with the presence of water will be catabolized by lipase into the single glycerol to release the three free fatty acid. All right. So this triple esterification reaction between glycerol and three molecules of stearic acid, 18 carbon without double bond. So in the presence of water, right, the glycerol, right, with the presence of three fatty acid, okay, it will bind to form triester of glycerol and release three water molecules, right? This is called esterification or condensation, right? Or dehydration synthesis of a triacylglycerol, right? So next is another reaction, we call it hydrogenation, right? Where the polyunsaturated fatty acid, for example here, polyunsaturated with two double bond with reaction with hydrogenated, it cause the polyunsaturated become saturated fatty acid. So the double bonds carry a slightly negative charge and readily accept positively charged the hydrogen atom, so it creates a saturated fatty acid. So this is often caused or present, or the process is occurred in fat as a partially hydrogenated. So usually this process will cause the cis configuration of the initial structure will cause into, or it will create trans fatty acid. Okay, so this is unsaturated, right? The process through iodine, it will also cause saturated, where the iodine will insert into the or attack the double bond, it become iodination. So it's also considered as a hydrogenation, and it forms saturated fatty acid. So we look again on the hydrogenation, where cis of the nature or naturally occurring fatty acid will transform into trans fat. So in nature, as I mentioned earlier, most double bonds are in cis, meaning that the hydrogen next to the double bonds are same side. But when it's undergo partially hydrogenated through a process in the factory, for example, to form margarine that's spreadable. So some of the bond will change from cis to trans. So just to give you a quick review on what is cis, what is trans. So if you look on the left, so this is a malic acid. So we look on the structure of carboxylic COOH, which is on the same side, right? However, for the fumaric acid, so we can see that it is on the opposite, right? So for trans fumaric, so the C double bond COOH on this carbon at the bottom is on the left, while carbon on the upper part is on the right, right? So it same goes for the cyclization of the structure. If it cis, it's on the same side of the structure. However, if it's trans, it's opposite, right? So this compound with double bonds or rings, observe attached group lying above or below plane of double bond or ring. All right, so cis we consider as group on the same side, and trans is group across, or as I said just now, opposite, right? So the different isomers have different physical and sometimes different chemical properties. Okay, so on the left hand side, to make you more clearer on cis, you see this is how the double bond in nature causing the bending of the structure of fatty acid. Remember when after undergo hydrogenation, the fatty acid will transform into trans fat. So it looks like almost a straight structure, right, of the fatty acid. Okay, just to share with you two differences between cis of oleic acid on the upper part, right, and trans oleic acid on the structure below. So what is the difference between cis and trans? So almost all, as I mentioned many times, the unsaturated fatty acids in the form of cis stereochemistry. So small amounts of trans are produced in stomach in some ruminating animals by partial enzymatic hydrogenation of the polyunsaturated fats. So it does present in small amount in milks and butter. So in factory, so this is what happened where the commercial hydrogenation of fatty acid from cis, usually when we extract oil that in the form of cis configuration, which is a good unsaturated natural fatty acid. But if since we want to change it from liquid into partially margarine that's spreadable, so that we want to make it a bit solid, we have to do a hydrogenation process. So when we add hydrogen through this partial hydrogen catalyst, so the unsaturated fat from cis becomes saturated, which is bad, and then it becomes worse when it transforms into trans unsaturated or unnaturally bad, right? So this is again a structure of triglyceride. For example, oil, cooking oil, which is after we isolate from a plant, and this is what happened when we use uh, oil for cooking. So, for example, when we use the first time for cooking, so only one of the fatty acid may be transformed into trans. So when we repeat the second times of using the same oil, so it will cause the second fatty acid to become trans, and later all of the three fatty acid in the oil become trans, which is that's why it is recommended that we recycle our cooking oil 3 times, not more than that, right? So you can also see the difference after you utilize so many times through the color, right? Through the characteristic of the oil become solid after you have utilized it more than 3 times. All right. So, an unwanted byproducts. So, other than talking about this trans fat, this it's also cause a bad to health due to it can cause to increment of the low density lipoprotein in the body. So we call it LDL. So that's it deemed to be heart unhealthy since the LDL tends to deposit cholesterol in the arteries rather than transport. Okay, so the good lipids in the body, we call it HDL, high density lipoprotein. So if it's our body system, which a lot of trans fat, it will cause our LDL increase, right? Which can cause what we call this atherosclerosis, right? Blockage of the arteries. So just want to share with you example of some food labeling. So this is considered example of incomplete labels. So it doesn't show trans fat information in the label, right? Only energy, protein, fat, but it's no trans fat, right? Similarly to the left, right? So this is example that showing misleading label. So you can see that it show several fat, polyunsaturated, monosaturated, right? Same goes to this one that show also polyunsaturated and mono, but doesn't mention trans fat, right, in the food labeling. And this is considered informative label, right? So it label the trans fat in gram, right? So so this is what is being the FDA wanted, that every manufacturer of food need to show the level of trans fat after the food had been processed, right? And this is another example to show that the trans fat is also not clear here, that mentioning it is 0 gram. So saying that this food has no trans fat. So I'm quite skeptical that if this what we call this potato chips, I'm sure there is using some oil, right? So must be there is some trans fat that present in the what we call this potato chips. All right. So now we look on what is fats and what is oil. So fats, we understand they are solid at room temperature and mostly they are saturated, and usually they are from animals, which is warm-blooded organism that fluid at warmer temperature. So for oils is basically they are liquids at room temperature, and most of it they has mono unsaturated or polyunsaturated fatty acid, right? So basically isolated from plants, and more kinks in the change, the more fluid the oil. So which makes the cell membrane flexible and permeable, which allow the nutrients and the waste product out, right, from the system, right? So the body fat is stored energy, okay? So we consider as fat, which is actually can be converted through unused carbohydrates, proteins, and triglycerides, which that make up our macronutrients into small globules of fat. So it is talking about how we consume excess carbohydrate and protein as well as triglyceride when we keep it in the body, especially in the adipose tissue, right? So 1 pound of adipose tissue can store around 3,500 calories of energy, right? So the high energy density of fat is ability to store energy around 9 calories per gram, right? So as compared to the carbohydrate and proteins is around 4 calories per gram. So where it allows us to carry or stores of energy with us. So that's why lipid is as a long-term storage. Okay. And it is also give for humans and other animals to the mobility and freedom to survive, right? So this is an example of camels. When we see the hump of the camels is actually containing triglyceride. So when they need energy in desert, so they can convert the triglyceride that present on their hump with the presence of oxygen to release in the form of carbon dioxide, water, and zillion calories, right? So again, fat is an essential energy for long-term storage in the form of body fat, triglyceride. So short-term energy storage from one meal is actually, as I mentioned earlier, usually come from the glycogen, which is we keep in our muscle from carbohydrate, right? So the adipose tissue is considered as a cushioning shield. So we need some fat, right, for cushion to shield our major organ like liver, the our what we call this, the our heart. So there's an adipose tissue around these vital organs. So other than that, it's also help to protect the organ against damage, right? And also physical shock. And also it's underneath our skin is provide insulation to our bodies, right? So and also guarding against rapid loss of our body heat to the external environment. So fats carry the flavors and vitamins of many of our foods, although fats have no flavors of their own. So it carry like vitamins A, D, E, K, which is vitamins that dissolve in lipids from our foods to our tissues. So fatty acid form not only the triglyceride, but other compounds as well, so including the prostaglandins and phospholipids. So just want to share with you, this is a picture of our adipose tissue, right? So all this round pink color globule is the adipose tissue, right? It contains the triglyceride. All right, now we move on to another type of lipid, which is polar lipids. So we have seen the non-polar lipid in the form of fatty acids, right? Which is fat and oils, right? Solid and liquid. So now we move on to look on lipid, which is polar, where it is considered as amphiphilic. It has dual nature. It has both the non-polar and polar structure. So the example of polar lipids is phospholipids, where it's allowed them to combine with protein molecules for the construction of biological membranes, where it's around the cell and cellular organelles. So we are talking here polar lipids as in the form of membrane as a permeable barrier that selects some molecules for passage but not others. So basically, a membrane, they are bilayer, meaning it has two monolayers or sheet of polar lipids. For example, glycerophospholipids and sphingolipids. So what are phospholipids? So basically it is similar to triglyceride in structure, except it's only have two fatty acid combined with choline instead of glycerol. So some example of phospholipids in foods are lecithin, egg yolks, soybeans with German peanuts. And this is structure of phospholipids. So this is the phosphatidic acid, right? And which is considered the polar head or charge, right? And this is the considered the non-polar tail, right? Or the hydrophobic part of the phospholipid. Okay, so we already see the triacylglycerol in fats and oils examples, right? Which is considered as storage lipid, non-polar. So B is the membrane lipids, considered as polar lipid, where it has example here, two types, glycerophospholipid, where glycerol bind with phosphate and alcohol together with two fatty acid, or in the form of sphingolipid, where sphingosine bind with only one fatty acid, bind with oxygen and any type of functional group that because the SX, right? So this is to show the structure of phospholipid, right? Which it combined with unsaturated and saturated fatty acid, palmitic and oleic, where the X can be any structure, right, of this glycerophospholipid. Okay, if we look on the list, if the X is hydrogen, we call it as the phosphatidic acid. If the X is ethanolamine, is considered as phosphatidyl ethanolamine. If the X is choline, we call it as phosphatidyl choline, and so on. Right? And now we look on the sphingolipid. So this is the main skeleton structure, right? So we look on the next table where if the position of X is containing hydrogen, we call it ceramide. If it's a phosphocholine on the structure of X, we call it as sphingomyelin. If it's glucose, we call it glucocerebroside. And if it's complex oligosaccharide attached to the X, we call it glycoside. All right. So these phospholipids functions part of cell membranes, which acts as the emulsifier, which help keep the fats in solution. So phospholipid is not a dietary essential, but it made by the liver. Right, so this is the phospholipid bilayer. So you have seen the head and tail. So basically the head is hydrophilic and the tail is hydrophobic. So meaning the head will be facing outside the cell as well as in the inside of the cell, cytoplasm, and the tail is facing inwards towards each other, right? So phospholipid bilayer or the cell membrane or the cell wall, right? So this is how the structure looks like. So we understand that the head is a hydrophilic surface. So in the middle is hydrophobic interior of cell wall, and the inner part is the uh, cytoplasm inside, water inside cell, right? So this is the phosphoglyceride aggregate cell wall. So this is the cell exterior and cell interior. So you can see the phospholipid, the membrane is aggregated, right? Where there are others that attach inside the membrane. You can see there is a glycoproteins, glycolipid, oligosaccharide, right? That bind within this membrane. Okay, we look on the first example. Lecithin is example of phospholipid, which is considered as emulsifying agent in eggs that usually we use in mayonnaise, in candy, and cake dough, right? So it has polar head and also non-polar tails. And this lecithin has two fatty acid, right? Where it has a charge on the structure of the choline containing and the nitrogens that balanced by a negative ion, right, using chloride. So it has a glycerol, right, by the phosphate and the choline. Right, so now we look on other type of lipids. So the first one is sterols. So from the name, we know it's cholesterol, hormones, bile, and also lipid-soluble vitamins. All are fall under sterols because of their structure. Skeleton structure are like cholesterol. And then we look on phytosterols, which is cholesterol found in plants. Examples, stigmasterol, beta-sitosterol, and camphorol. Terpenes is also considered lipids that present in plants and animals. For example, limonene, beta-carotene, gibberellic acid, squalene, and lycopene, which usually it provide colors and odors associated with plants. Next, we look on eicosanoids, which is short-lived hormone-like activities, which I shared earlier about prostaglandin, thromboxanes, and leukotrienes. And lastly, pheromones, which is chemical signals that release from organism into the environment that alter the behavior of the members of the same species. So basically, pheromones, they are usually used for sexual attraction, trail markings, and defense alarm for natural repellents. So we look on the first one, sterols, which is the structure consists of carbon rings. So you can see cholesterol, they have 4 carbon ring, 3, 6 carbon ring attached with 1, 5 carbon ring. So on the upper part is cholesterol structure and below is vitamin D3. So you can see that vitamin D3 is the structure is more or less looks like cholesterol with the open structure on the second 6 carbon ring, where it has only 2 6 carbon ring and 1 5 carbon ring, right? And the most important sterols are sex hormones, for example, like testosterone, vitamin D, biles that aids in our fat digestion, the adrenal hormones, the cortisol, and the cholesterol itself in foods that made by the liver. also dietary sources include in egg yolks, in liver, meats, and also in dairy products. All right, so this is a cholesterol that we know it is not good when it's excess in the body, which can cause atherosclerosis, right? So we know that cholesterol are high in some of these food sources, such as in red meat, egg yolk, and dairy products. So it is low in some of egg whites and yogurt. And of course, none of cholesterol present in fruits, veggies, and vegetable oils. So basically, cholesterol only come from animals. Right, so cholesterol is absolutely essential. So just now we said that it is not good in excess, but we still need cholesterol. For example, like male, average of weight 80 kilogram, so you need around 220 gram of cholesterol. For 50% in cell membranes, 40% in to convert it to bile acid, which is needed to emulsify your lipids, right, in the liver, gallbladder to intestine, and some of the cholesterol is needed to convert the hormones, right, to form testosterone, estrogen, and cortisol. And you can see the composition of the cholesterol in some of the organ in the male. For example, in blood, they are around 60 gram, muscle 45, in brain, they are 50 gram, adipose tissue 45, and so on. Right? So, and this is an example, the structure of cholesterol. So the precursor to synthesize cholesterol in our body system is through isoprene to form estradiol, which is estrogen that present in women, testosterone in men, cortisol as considered as our anti-inflammatory hormone, folate and glycofolate is our bile salt, right? That help to emulsify lipids in intestine. And then there are phytosterol, means sterols that are synthesized in plant in the form of stigmasterol, beta-sitosterol, and camposterol. So if you search about these three phytosterol, they are having good medicinal properties that had been studied or had been used to treat cancer patient, where it has very potent anti-cancer properties of this phytosterol, right? And then these are example of other lipids in the form of terpenes, right? Because of its nature, they are not solubilized in water. So, for example, like limonene, beta carotene, gibberellic acid, squalene, and lycopene. All these are giving a color, right, for the plant. For example, beta carotene giving a orange color to the carrot, lycopene giving a red color to tomatoes, right? And then just to want to highlight again about the cell membrane, where the cholesterol are embedded in our cell membrane as well. So how this cholesterol is transported in our body system? So we know that cholesterol is insoluble, right? It's not soluble in water. So they need a transporter. So the transporter of cholesterol, the main is the high density lipoprotein, or we call it HDL, or we consider as good cholesterol, right? That transport cholesterol in the blood. And another one is LDL, the low density lipoprotein, and VLDL, which is the one that deposits cholesterol on our arterial wall, which is the bad cholesterol. All right. So this lipid transport, there are basically 4 types where it clustered in the form of lipoproteins. We call it as chylomicrons, very low density lipoprotein, VLDL, low density lipoprotein, and high density lipoprotein HDL. right? So the size of the composition of lipoprotein, which it will be based on the density of protein. So for example, chylomicron, we can see that the triglyceride is the highest composition, right? And the very low density, we can see the triglyceride is less, more proteins that present than chylomicron, but you can see the most high density they have more protein, right? So meaning that's why HDL is good cholesterol carrier, right? So the structural features, HDL versus LDL. So we know HDL is the good cholesterol carrier, which contain unsaturated cis fatty acid, while LDL mostly they contain saturated and trans fatty acid. So these both contain apolipoprotein and phospholipids, which are oriented with hydrophobic part on inside and out the hydrophilic edge, which is exposed, right? So next is the cholesterol that in the gallstones. So we consider it as a bile that used in our fat digestion. So this bile is contain water, cholesterol, bile salt, proteins, and bilirubin that as the waste product. So if bile contains too much cholesterol, so it can harden into gallstones. So the extraction of cholesterol from gallstones will have to be removed through surgery. So this is a common lab experiment that being uh, done right in the lab. So the size can range from grains of sand up to golf ball. All right, so this is how the emulsification of fat by the bile salt is occurred. So for example, the fat, and then there is a watery, the gastric intestinal juice, and there is an enzymes, right? So the enzyme in the gastrointestinal juice can't get to the fat, right? So when there is a bile, right, enters the small intestine, so bile has an affinity for both the fat and the water. So it can bring the fat into the water. All right. So once the fat is emulsified, so the biles now can become the make the fat become a globules into a small droplets that repel each other, so that later the enzyme can react, right, and digest the what the fat, right, in the intestine more efficiently. All right, so you need to understand the enzyme is soluble in water, so the bile is trying to emulsify to make the fat dissolve in water, so that the enzyme can digest the emulsified fat, right? So the source of bile acid usually produced in the liver and is stored and concentrated in the gallbladder, secreted into small intestine. And then if the gallstone block and exit duct, then the gallbladder may need to be removed. So the fat digestion become more difficult, but it's not impossible. It can still be removed, right, through surgery. So this is an example of um, hardening of gallstones due to high cholesterol level, right? And what are the risk factors of the gallstone? So for example, like women, they are 2 times likely have the risk of excess estrogen from pregnancy, birth control pills, and hormone replacement therapy. So it may cause increased cholesterol levels, right? That can increase the risk of gallstone formation. and maybe due to the obesity, due to high fat diet or low fiber diet can also cause the gallstone formation, or rapid weight loss, which is caused by crash diet, due to the body tries to metabolize fat, or the age is more than 60, so body will secrete more cholesterol into bile. And this is to show you a list of lipid content in a milligram per serving of common foods. So the list will show you the cholesterol level, the saturated fatty acid and unsaturated fatty acids. For example, beef, right? We can see the cholesterol is around 91, right? And the saturated 2.7, and monounsaturated is 2.7, polyunsaturated 0.5, right? Butter is 219 of cholesterol, saturated 50.5, monounsaturated 23.4, and polyunsaturated 3. So if you go through that, we can see eggs, the whole eggs, the showing the most high cholesterol, right? If it's not omega-3 eggs, right? And then you can see among this list, the most saturated fat, we can see the highest is in the butter. The mono unsaturated, the highest we can see in the margarine, right? And the polyunsaturated, the best is the safflower oil, right? So just to share some information here. So when you go to the market to buy palm oil, so this label is also misleading, right? We know that palm oil is come from palm tree, right? So, or the palm seeds. So palm is plant. So we understand that plant, they don't have cholesterol. So just to attract users, right, to buy palm oil, they just put tanpa cholesterol, which is basically, of course, palm oil is a plant, so plant don't have cholesterol, right? But it has others, either saturated or unsaturated fatty acid that present in the palm oil, right? So we can see that palm oil has like around quite high saturated fatty acid, around 49, right? Also some monounsaturated 39.2, and also polyunsaturated fatty acid, right? So the kernels is also now being used to extract the oil. So it has a high in lauric acid. All right, in total, the saturated around 82%, right? So yes, I already highlighted just now. Note that no cholesterol is produced from plant sources, right? And to just highlight again that the vitamins that soluble in water, they are considered fat solubles, so they are considered under category of lipids, the vitamin A, D, E, K. All right, so cholesterol related compounds, the vitamin D, that usually we have to get it from sunlight, the 7-dehydrocholesterol. And this is the process where we synthesize vitamin D. So when we got exposed to the sunlight, so the 7-dehydrocholesterol will be converted to cholecalciferol to become 25-hydroxycholecalciferol, and lastly become the active vitamin D, right? So are you getting enough vitamin D? So basically, if you are deficient of vitamin D, it will lead to poor teeth and bone development, or we call it rickets in children, right? So so we can get vitamin D from milk, or from the sunlight, right? So if we have deficiency, so for example, like people who are staying in around winter where there are like sunlight is lacking, so they most of them may have a problem in calcium absorption. All right, and the darker skin also can prevent the UV action, right, which may prevent them to absorb the vitamin D, and other than rickets. So low vitamin D can also cause or increase the risk of getting the multiple sclerosis, right? So can one get too much of vitamin D? So overuse of vitamin D supplement can lead to excessive absorption of calcium and formation of calcium deposit in kidney, right? So exposure to sunlight is not a problem. So because we have melanin, so it only can cause tanning to our skin, right? So other than the vitamins, so you need also to know that eicosanoids is also lipids, which initiate the inflammatory response, pain and fever associated with injury, right? So example like cuts, scrapes and blister, which is at the end we cause chronic diseases like osteoarthritis and rheumatoid arthritis. And this icosanoid is important to promote blood clotting process for the platelet aggregation and regulate blood pressure. For example, the prostaglandin for vasodilators. And then it's also help to control reproductive function. Example, induce the labor, cause the contraction, and also for role in male fertility. And it's also regulate temperature in our sleep-wake cycle in animals, also in human, and also promotes muscle contraction. And this will be the last part of the topic where I would like to share with you some fat substitutes that currently available in the market, where it's either carbohydrate based, for example, oat trim and zec trim, which is made from plant fibers, or protein based, or we call it simplis, which is made from egg white or milk protein, and the one that is fat based cholesterol, which is made from sucrose and fatty acid, which had been shown to pass through body undigested. Right. So these are example of fat substituent. So if you can't get fat, so you can get the substituent from this kind of products, right? So there is a lot of questions, queries about the fat substituents that now available in the market. Do they work? For example, the first question, is it giving a potential health benefits like other fat? Is it promote weight loss and lower our blood lipids, right? Um, but like sugar substituents, many people just eat more instead of replacing fat. And there is there possible side effect of these fat substituents? And for the moderation, so how far that this fat substituent can do their work, right? So I leave it all these questions. So you may search further about the example of the three fat substituent that I have shared, right? And these are some exercises that you can do right before your test. So these are the first question or the first exercise. What is the function of triglyceride, right? And then question number 2, which type of lipid makes up the majority of the cell membrane? Question 3, fats that have at least one double bond between carbon atoms are called, And exercise 4, phospholipids have a. And the last question, which is exercise 5, which of the following is an example of a steroid? All right. This maybe the last slide. Okay, thank you guys.